She funded in two.
So much for the invitation and the opportunity to speak to you. It's great to be here. So for this talk, I will outline what I mean by confounder-aware systems medical approaches by introducing the overall idea and goals of that kind of research project, mostly by giving you examples from some past and currently ongoing work we're doing. And the goal of that and the goal of the discussions we can have thereafter would be perhaps not so much that the results are interesting, but that you might find some of the methods or approaches to be useful to you in work that you want to do. So as mentioned, my lab is working on host microbiome interactions. And in this, I'm particularly interested in how it is that microbiota that exist inside or on a human host tend so often to persist in composition or activity over time within that individual, even though it varies a lot between different individuals. This indicates that on some level, the human host is able to control its microbiome composition, and we believe that the immune system must play an important role in this, but also that this might be something which is bidirectional, where, for example, early exposure to certain colonizing bacteria would imprint on the immune system and cause tolerance for them while avoiding other types of bacteria, as well as other forms of microbial immunomodulation, such as, for example, pro- and anti-inflammatory activities of different bacteria. Moreover, this type of interaction seems to be potentially relevant in a number of different states of health and disease, many of whom have an immune system component in place as well. This includes metabolic diseases, cardiovascular diseases, inflammatory and autoimmune diseases, as well as some psychiatric diseases, most likely, which, even though this is a very hyped field and a lot of what we are publishing or what people are publishing probably won't validate, a lot of it I think will, and we'll probably find some therapies out of this that can be used to improve management and prevention of some of these diseases. So we want to try to chart to what extent the microbiome is an integrator of environmental signals in causing different disease phenotypes, to what extent the immune system acts in turn as an integrator and mediator of microbial signals, and to what extent any of these processes depend on different small molecules in circulation or in specific tissues, including pro- and anti-inflammatory microbially produced or metabolized compounds. And the way that we want to try to do this is ideally in a hypothesis-generating or primarily hypothesis-generating high-throughput manner, starting from cohort data whenever possible. So even though we will work also with animal experimentation, which allows certain interventions and perturbations otherwise not possible, the best is to be able to start with actual people, because in the end that's what we want to get back to, where we want to be able to try to improve diagnostic, prognostic, or therapeutic approaches. So generally, this kind of multi-omic study would involve either a cross-sectional cohort where you look at sick versus healthy people or people at different stages of disease, or better, longitudinal cohorts. I'll return to both of these examples later, where you can follow the same individuals over time as disease progresses or reverts or is treated, comparing different interventions, and thereby circumventing a lot of this high individual variability that I mentioned for these features. So it will start with patient samples being part, in our case, typically as a clinical collaboration, gathering as much relevant biosamples as possible, both for microbiota, including mostly the gut microbiota, but also lung microbiota, oral microbiota, skin microbiota, increasingly more and more. Host samples, easiest to get, of course, is things like blood samples. In rare cases, we could get access to biopsies, but that's typically only when there's a surgical intervention anyway going on. And what we would call metadata, but which could more aptly perhaps be described as non-omics data. So various standard clinical readouts, demographic risk factors, treatment regimes of these subjects. And then comes the fun part, or no, not quite yet, but we quantify all biosamples generating data in whatever way is accessible. So for immune system quantification, there are things like FACS cytometry and gating cytometry, and increasingly we're looking into sequencing-based approaches to study populations of immune cells the same way that we would study populations of microbes using sequencing and a metagenomic approach for understanding the microbial communities with regards to their composition or their functional activities. And for metabolomics, proteomics, glycomics, there are also various platforms, most of which we do not actually have the capacity ourselves to do, but where we have collaborators and core facilities that can help us. Then getting to the fun part as a bioinformatician, namely having all these data matrices and trying to make some sort of sense of them and model the interaction between the different systems of the whole organism under study, the human and its microbiota, under whatever intervention or state change is happening, bringing into bear also previous data from other comparable studies, as well as whatever data we have from evolutionary analysis, for example, to understand the functional roles of the host and microbiome components in this context. And this can be either ranging from very simple to very complex, starting with simply statistical tests of significant differences or correlations between features, to trying to work out what the causality is and building networks of directed edges, linking together measurable components within the system of host microbiome whole, leading from external intervenable circumstances to a health phenotype. In the end, of course, this is still association hunting, and one would not base any treatment decisions merely on this at this point, but we'd proceed to somehow try to validate key findings with experimental collaborators, for example, testing if potential pathogens have that effect in a model system, or if potential interventions have the expected impact on health and disease before they can then be brought into translation and improve, hopefully, eventually, treatment regimes. So I'll go into some examples of how we've been trying to get towards this kind of research program, which will be most of the rest of the talk, to give you examples. I'm focusing in this on the non-straightforward aspects, which are how to deal with confounding influences. And by confounding influences, essentially I mean that beyond the fact of merely features that are involved as causative factors of a disease, meaning they are potential treatment targets or potential prognostic diagnostic targets, there are also other potential causality scenarios happening. You have things that emerge downstream of one or more disease phenotypes, which are an indicator, but perhaps an unspecific indicator of the disease phenotype. Can also follow from other similar situations. You have features that are purely indirectly associated as they share a common cause with some risk factor. And in human populations, as opposed to animal model systems, of course, these are extremely multifactorial diseases that combine essentially out to the level on human societies, how risk factors integrate into affecting the individuals and leading to disease. And last, whenever working with patient data, we must consider the fact that these people are actively under treatment for their diseases, meaning that the medication regimes, if they have a competent physician, will be adapted and changed until the phenotype has been controlled as well as possible. In particular, in cases like cardiovascular disease, you have people on a lot of different treatments at once, and that's only the pharmacy. There's also factors such as lifestyle change resulting from drug treatment or resulting from diagnosis and advice being given by physicians, each of which in principle could also be side effects. And the examples I'll give will in part involve how to try to circumvent the risks that we draw the wrong conclusions from what is actually a downstream treatment effect rather than a cause of a disease state, But perhaps most interesting would be cases where we can identify the mechanism by which a treatment operates so that we could effectivize it. For example, are there cases where a drug is mediated by a change in the microbial ecosystem in the gut, in turn changing metabolic state and in turn improving disease phenotype? This would imply that we could try to target that microbe system more directly and improve treatment further. So some examples I want to give are from the MetaHit project. That was the first large-scale European gut metagenome analysis project, so looking at the human gut microbiome through metagenomic shotgun sequencing, primarily in metabolic disease, so insulin-resistant and diabetic patients, but also in inflammatory bowel diseases, and try to find biomarkers of these diseases that could help improve treatment. So one of the things we looked at when this happened was the composition of the gut microbiota. What you see here currently on the screen is a projection down onto 2 dimensions of the composition of the gut microbiota in terms of which bacteria are there, in what proportions, in subjects from, in total, 3 different populations, a Danish, a Swedish, and a Chinese cohort of type 2 diabetic patients and healthy controls, further split up depending on whether or not they're receiving what at least at the time was the most common anti-diabetic drug, namely metformin. Initially, when we compare these data sets, we found a large discrepancy between how the microbiomes looked within the data that made it seem as though the diabetic signal was different between the data sets. As we looked closer, we saw that there was a difference between these cohorts in how many of the diabetic patients were medicated with metformin or not. And when accounting for that, what we see in this figure is in fact that the degree of change in the microbial composition in the gut of the diabetic patients depends much more on whether or not they're metformin treated than whether or not they are diabetics compared to non-diabetics, type 2 diabetics in this case, which was further then validated in a longitudinal and animal setup by our collaborators in Gothenburg. And this raised the question, if there is this kind of effect just with one single type 2 diabetes drug, what other effects might there be there that we should need to control for if we're trying in human patients as opposed to experimental animals to understand the actual role of different gut microbial components in the pathology. Just to further exemplify here, we see that in particular there's 2 taxa that were in this study connected to metformin treatment, in that metformin-treated patients had higher E. coli prevalence and abundance in their gut, and lower prevalence of another bacterium, which is Intestinibacter bartholeti, which again, we could show that later by giving people metformin, following them for some time, and then seeing the changes happening, and also seeing that those changes contributed to some extent to an improvement in glucose control, which could be transferred also to a mouse phenotype by our collaborators in Gothenburg. Moreover, when controlling for this and adjusting for the metformin treatment, we can see a underlying phenotype or an underlying signature of gut microbial changes, mostly depletion of bacteria that produce anti-inflammatory short-chain fatty acids in the gut of diabetics compared to controls, which is furthermore not shared between type 2 diabetics and type 1 diabetics, which is a very different etiology of the condition, therefore indicating that this is probably a causal factor rather than a downstream factor. Again, this is an example in the case of one disease of how it's necessary when working with human data to try to account for the treatment effects that will be present in the cohort when searching for biomarkers and mechanisms for disease. After MetaHit concluded, another consortium started called Metacardis. It ran under EU funding from 2012 to 2018, and we are currently publishing the first papers to come out of this, which was intended as a successor project for MetaHit. It's using in total 2,000 people from Denmark, France, and Germany at a range of diseases going from healthy through various degrees of metabolic syndrome, so increased hypertension, increased dyslipidemia, increased insulin resistance, into gradually higher obesity, type 2 diabetes, and then going into the cardiovascular part, so acute coronary artery disease, chronic coronary artery disease, and finally heart failure, and trying to use this as a sort of cross-sectional equivalent of a longitudinal study to understand what the specific and general biomarkers for these disease states would be, both within the gut and within factors such as serum and urine metabolite spaces, trying to find new diagnostic tools, new prognostic tools, and so forth. Knowing what we did from MetaHit, we were considering the extent to which these people were medicated. So the heart failure patients can have up to 8 or 10 medications at once, some of them medicated, some of them managing the side effects of other medications, such as taking protein pump inhibitors to mitigate some of the other side effects. So we knew we needed some way to account for these effects in looking for the disease-specific biomarkers. Just as a brief view of this, if one looks at the overall structure of the measurable spaces of the gut microbiome composition with regards to its taxonomic composition, regards to its functional profiles in this cohort of 2,000 people, or in regards to its antimicrobial gene resistance gene carriage profile, as well as the chemical composition of serum and urine metabolites in this data sets, we can try to break down what you see here, to what extent the individual variability of these subjects can be explained by different factors that we can trace in the cohort. We see that a very large fraction of the variability between individuals is traceable to one or other of the types of drugs that they are given, which includes both their history of antibiotic use, which I find super, super interesting, but also their current regime of anti-diabetic, antihypertensive, anti-lipid medication. And this influence is in many cases larger, as we saw in MetaHit, than the influence of the disease itself or on other known environmental factors such as diet. So this is very interesting, I think, and we try to proceed there to see, can we somehow filter out these potentially confounded signals, or at least these covariates which are not disease biomarkers but rather treatment markers. So we've developed a software package which now exists in a beta version. It's not online yet, but it will very soon be, and we'd be happy to have more people then eventually use it, which is a way to do searches for univariate biomarkers of a disease in a cross-sectional analysis of features that distinguish, for example, sick from healthy, while taking into account a large number of possible covariates, and doing so at a large scale, so potentially thousands of microbes or thousands of metabolites, or for that matter, differentially expressed genes or any other such feature. Essentially looking at, for each apparent possible signal in just a naive test of its association with the phenotype in question, what other covariates also possess that association. When we have that matrix of potential confounders, we can do something which is essentially a model selection problem. So trying to, for each potential feature covariate pair, look at the two potential different explanations, either that there is a confounding influence or that there is not, and then see which model provides the best fit to the data. And this can also be done in the opposite direction as well. Going from this, then, we can try to classify the initial apparent biomarkers into three different categories. So one is where there's no alternative explanation from any of the tracked covariates, which could be things like being given this medication or that medication, or etc. Cases where there definitely is some covariate present in the data that's a better explanatory factor than the disease status itself. This would be examples like the metformin versus E. coli signal we saw before. And then a middle ground where it's really impossible to tell because the features are so correlated. Examples of this being that heart failure patients will virtually always be on a combination therapy of beta blockers, aspirin, and statins. So we cannot really say what is a pure heart failure signal versus what is a signal of this triad of medications. But at least this allows the separation of the initial apparent features into different sets that can be then examined further. So just as a few examples of what this yields, and again, hopefully we'll resubmit this sometime this year and get it out before we are scooped by our competitors. Just again, looking at type 2 diabetes as an example, there's about 6,000 measured associations in microbiome and metabolome in the Metacardis cohort between type 2 diabetics and healthy controls, out of which about half of them can be deconfounded in one way or another, so we can classify them as having some direct association to their disease phenotype, whereas the remaining parts are traceable to one or the other of our treatment confounders or other confounders, including features that are actually rather properties of tobacco use, features that are traceable to antibiotics taken in the last 5 years, or to any of the medications that the subjects are on, largely in the case of the type 2 diabetics from their anti-diabetic medication, which makes a lot of sense. And we can further link together different drug classes on whether or not their influence on the microbiome and the circulating serum and urine metabolome seems to be unique or shared, where the really interesting part comes in whether or not drugs that share an association to a measurable feature then might be operating along similar pathways downstream in the organism to cause their therapeutic effects. And here also, of course, it's notable, we cannot always be sure on whether these features, these associations between a treatment and a measurable feature are direct effects. Some of them are likely to be, and some are likely to actually be severity markers. So they're found more extreme in medicated subjects because those subjects that have this more severe phenotype are the ones that require this medication to be able to reach a stable state, and their physician will keep on trying medications until they reach that. So this I hope to be able to show more on at some point later this year. I'll also note that when we do this, we don't throw out all expected signal, but we do see a number of previously reported features emerging. For example, all the different related pathologies that I was talking about, the metabolic diseases, the cardiac diseases, etc., share an underlying signal. For example, in case of elevated capacity for the microbiome to produce branched-chain amino acids, depleted capacity of the microbiome to degrade branched-chain amino acids, an enriched carriage of branched-chain amino acids in serum, indicating a potential causal role of the microbiome in causing insulin resistance and therefore driving these diseases. We see a similar phenomenon with lipopolysaccharide production, which is consistent with the idea that these metabolic diseases in part stem from pro-inflammatory bacterial compounds penetrating through a leaky gut into the bloodstream and causing a state of sustained low-grade inflammation, and an inverse relationship where short-chain fatty acids, which are anti-inflammatory, their production is depleted in the subjects that are suffering these diseases more often, and the capacity to degrade them is sometimes higher, and the carriage of these anti-inflammatory compounds is likewise lower. So it's in line with what we've been suspecting, and we are again hoping to make a lot more of this soon. I'll move on to another example of confounding influences. Aside from the treatment factors, there is the problem of technical factors. So depending on how you do your measurements in a high throughput setting, for example, in the case here of microbiome analysis, it depends a lot on how you process your samples, what kind of results you will get out, because different methods for extracting DNA from a microbial community, for example, might have different efficacy for different bacterial taxa. So we wanted to compare different working pipelines in the lab for handling, in this case, stool samples for gut microbiome analysis, using existing methods, including the gold standard of keeping samples in on dry ice and in -80, compared to techniques such as stabilizer solutions, as well as different extraction kits, to see will the potential influence of these confounding factors, these technical variability factors, be large enough that they will look like biological signals and therefore potentially cause false conclusions, especially in studies that are not fully homogeneous with regards to a study protocol like this, which unfortunately happens more often than we would like, because often we are forced to use one study as a control data for another study, given that it's still relatively expensive to generate this kind of sample. So we took volunteers, sampled them over time, and replicated the analysis using a variety of different DNA collection methods and the different DNA extraction methods, then the same underlying analysis pipeline, and using the software package MetaDeconfounder that I mentioned before to analyze it. So just looking at the initial output, what we see very clearly is that the method used for collecting and storing DNA does not have a super large effect on alpha diversity, so richness of the ecosystem in the gut that we recover and estimate, though there is some effect, and most methods have retained lower diversity than the gold standard of dry ice, so there's some loss of microbial signal with every other method, but the real difference emerges when comparing different extraction protocols for DNA from stool samples, where there's a lot of difference between two of the main protocols that are on the market. Moreover, if we look at the result of this in terms of large-scale structure of the recovered microbiomes, so you may recall that there is a concept of enterotypes, which is like blood groups for the gut in a way. So large-scale patterns that repeat in individuals of composition of the microbiome, with some people being high in Prevotella bacteria, some people being high in Bacteroides bacteria, some people being high in Firmicutes bacteria. All of this, of course, simplifying, but possible to recover as recurring patterns, and probably reflecting metabolic sharing and co-occurrence patterns between bacteria in the gut. We see that there is a major shift in where samples end up classified into enterotypes depending on which of the extraction kits for DNA samples. Again, these are both used in various studies. So if one were to merge together data from studies using different protocols, it would very much look like a difference in overall structure of the microbiome that has otherwise been associated with health factors. And this is sort of showing the same thing, that there's a skew happening between enterotypes depending on the DNA extraction method. There's also some skew happening based on DNA collection methods, though not quite as severe. One of the reasons we did this study was, of course, we want to make sure that we have the best setup possible for the studies we conduct to make them reproducible and comparable to other data sets. We believe that the main reason why this happens is that the different protocols differ in terms of the mechanical disruption of bacterial cell walls in a way that creates a skew if you have only one size of microbeads for breaking the cell walls, that essentially loses out on gram-positive bacteria, whereas the other methods are slightly better on this. So we can make some recommendations, but above all, we will want to note that this is a factor that has to be taken into account. When you design a study, you need to account not only for the treatment factors, as I mentioned, but you also need to account for technical variability and a coherent protocol, ideally, throughout all the samples that you take. Just looking at the output, so this is looking at the effect of extraction protocol in the leftmost column on particular gut bacterial species, their composition in this cohort, and then comparing that to the relative effect of different DNA collection methods, and comparing it similarly to the effects of other properties that could, in principle, affect microbiome composition in the data set, such as various demographic features, effect of nutrition, because we had these people undertake a food frequency questionnaire, And there are associations, which we also find in this data set, to different clinical parameters such as blood pressure, cholesterol, and insulin resistance. And the point I want to make with this is that there's overlap between features that are affected by the technical error sources or variability sources, such as the DNA extraction protocol or DNA collection method, and features which are truly affected by genuine biological features such as clinical health state, nutrition, or risk factor demographics, meaning that unless this is accounted for, we are very likely to not only draw erroneous conclusions, but erroneous conclusions that on the surface do not seem nonsensical. So another reason to ensure that the possible impact of the choice of protocol for creating samples, and this goes, of course, far beyond microbiome analysis. It would go into any kind of biosample management, or for that matter, other kind of data collection you do. It's important to consider the technical variability, because otherwise you will draw the wrong conclusions. Just as a few more examples, there are indeed features that either are detected only in the one or the other protocol that have previously in the literature been associated with risk factors of cardiovascular disease. Just underscoring the point I just made, that unless accounted for, this is likely to cause erroneous conclusions. The last study I want to talk to you about, I have no idea how am I doing with time, actually? Am I above time yet, or?
No, it's good.
Okay, perfect. Is a similar setup which combines some of these previous factors in a longitudinal study of intervention into subjects suffering from a metabolic syndrome, in particular suffering from hypertension. And I'm interested in longitudinal studies, particularly because a lot of the factors I just mentioned that you saw were different between individuals, including their habitual diet, including their background, including their demographics, etc. Those are not changing a lot within individuals over time, which means that in principle one can factor out a lot of the demographic noise and a lot of the risk factor noise by seeing what happens within a single person as some condition is altered or as time passes, which not only protects us to some extent from drawing wrong conclusions, but also gives us better statistical power, which is, at least in this kind of omics analysis, generally the limiting factor. If we could do 10 times more patients, we would have a lot more power to detect associations. But since we do not, we have to try to be as efficient as we can with the participants we have. So this was a randomized intervention done in subjects going to a clinic south of Berlin to deal with hypertension and metabolic syndrome by making a major dietary change. So both the control group and the intervention group shifts on to what's essentially a Mediterranean diet, the DASH diet, the dietary strategies against hypertension, with some modifications and also some other lifestyle changes happening in both of the groups, trying to ensure compliance to the protocol by having weekly sessions also where they're counseled on their progress, because this is known to be something that can indeed reduce hypertension to some degree. But the really interesting part was that one of the groups, before beginning their DASH diet, first went on a 1-week fast where they drink, I think, 500 calories max of red beet juice every day and otherwise eat nothing, which is kind of a fad diet in Germany and elsewhere. And we were curious to see if it could have some further positive impact. So essentially fasting changes to the homeostasis of the host and microbiome by performing these two interventions in tandem. So subjects were followed in total for 3 months. The study was called Corona, by the way, no relation to the currently ongoing pandemic. It's entirely unrelated. And they were sampled at baseline. They were sampled after 1 week of either fasting intervention or starting the DASH diet, and then after 3 months again. And at each point, we took stool samples for microbiome analysis. detailed phenotyping, including a lot of blood pressure measurements, but also other features, and then taking blood samples to do a cytometry analysis of the immune cell populations, because again, our hypothesis has been that there is some influence taking place by way of changes between pro and anti-inflammatory immune cell subpopulations somehow that plays a part in the pathology. So what do we see then happening? So one thing that surprised us was that in terms of alpha diversity, so overall richness of the gut microbiome, that actually didn't change too much on the fasting. We had thought it would, but it doesn't seem to do that. Nor did we see any change in overall beta diversity, and this might require a bit of explanation. Basically, there's something called the Anna Karenina hypothesis, which is from this idea that all healthy families are healthy in the same way, but all dysfunctional families are dysfunctional in different ways. And in the same way, there's more ways of being out of homeostasis than being within homeostasis. So one would expect somehow to see a greater variability between subjects after a perturbation like the fast than before the fast. But this also was not the case. What you see here is a violin plot of the distances between all the microbiome samples in terms of composition of their gut ecosystems. But we did see an effect in what that composition looked like. So here is the gut microbiome of the study participants in the group that underwent a combination of both fasting and switched to a DASH diet, projected onto the first 2 dimensions of our principal coordinates plot. And each subject is shown, there's 3 time points, if you recall, is normalized to the middle point. So the blue arrows, or the red arrows, represent the shift between individual starting point and after 1 week of fast. And the blue arrows going from origin to end point represent the subsequent shift from the 1 week fast state to wherever they were 3 months past the intervention. And what I want to draw attention to is that these 2 distributions of changes are not centered in the same location, even though subjects change in different individual trajectories and moreover from different individual starting points. There is a trend of shift that is characteristic to fasting, and there is a trend of shift that is characteristic to refeeding after fasting in terms of gut microbial composition. These 2 are largely opposed. So there's a shift towards a fasting specific stage, and then sensibly, I think, a reversal back towards the original state 3 months after. We see the same thing happening in terms of the immune system composition, so the relative proportions of different immune cells, Treg cells, Th17 cells, T1 cells, etc. That's actually even stronger or even more striking in this visualization, that the immune system shifts overall in its composition upon fasting, and then it reverts over the next few months back to where it was. If we look closer at what features drive these changes, we see, oh yeah, this is another important thing that we need to take into account. So the subjects were, as I mentioned, hypertensive metabolic syndrome patients. Many of them were already on some sort of antihypertensive medication. Most subjects could reduce, or many subjects could reduce their antihypertensive medication in the group that underwent both fasting and a shift to a DASH diet. Was a trend towards this in only a DASH diet, but not as strong, meaning that enough of them apparently had an easier time maintaining normal blood pressure with less medication, which is great in terms of implying that there's an improvement happening from the intervention, but it causes us a problem, because this is exactly what we try to avoid with the longitudinal setup that we have to control for a treatment confounder. So we needed some way to account for changes in potential covariates happening during the intervention. So for this purpose, we designed, or my student started designing another software pipeline similar to one I just described before, where it's actually analogous to it, where looking at the initial spontaneous associations when one doesn't account for changes in potential confounders, then try to see which features that are co-changing with the differences seen over time in the individuals followed, and then performing, again, model comparison tests to see, is there a better explanation? For example, is this change in a bacterium happening when an individual goes off a fast and refeeds again actually rather better explained as a result of them having gone off their antihypertensive medication, and then trying to filter essentially the resulting features out as being either confounded or ambiguously confounded, again analogous to the previous pipeline I described, or being possible to deconfound for these treatment changes. This, coincidentally, this is also going to be an R package that's out fairly soon, hopefully for people to use. We think can also be used as a way to try to identify mediating factors in an intervention like this, which is something we want to explore conceptually further, trying to find intermediate steps that could be used for more direct perturbation. Yeah, as I said, it's classified into potential different outcomes, and all the results that I'll show you here are the ones that pass this filtering for potential confounding influences. So if one wants to look at what specifically is driving this overall change in composition, so just looking, there's thousands of immune cells changing, but that's just a subset of what's happening. Broadly, one could say that it seems like fasting is a shift more towards dominance of the innate rather than adaptive immune system, and then under refeeding, the adaptive immune system again starts to have more influence, though this is an oversimplification. It ties in also to some specific intestinal immune cells that we want to test further. More interesting to me, I think, is what happens to the taxonomic composition of the gut microbiome, because it seems on the surface of it that a lot of the changes happening during the fasting, that's the red arrows here, is a depletion of bacteria that we know are associated with health, in particular short-chain fatty acid producer bacteria such as Eubacterium rectale, Coprococcus comis, Faecalibacterium, whereas there is an initial enrichment of features or bacteria that have been associated as potentially opportunistic pathogens, but then this pattern reverses upon refeeding, and does so more strongly. And the analogy I would like to make there is what happens when someone is trying to build muscle. They perform exercise that degrades and breaks down and hurts the muscle, but under proper circumstances, regrowth occurs stronger. And this is what I think is actually happening under the fasting intervention, that the bloom of bacteria once fasting ceases is enriched for commensal bacteria that can soothe the inflammation and thereby cause a shift in the immune system towards a calmer homeostasis, which is conducive to better cardiovascular and metabolic health. Looking at the functional potentials changed, I won't go into detail, but I will say that of particular interest here is an increase under fasting in propionate production, which is one of the short-chain fatty acids that we've also found in animal experiments and in another cohort to be associated with a better, a less hypertensive state and a better homeostatic state. So there seems to be at least two different scenarios happening here: some early bloom in propionate production under fasting, and then a later bloom possibly in butyrate and acetate production. As I mentioned, there was a change in medication happening, and even when accounting for this change in medication, the subjects that underwent the full treatment of both fasting and going over to a DASH diet, there was an sustained reduced weight loss and improvement in blood pressure, and that made us want to look closer at which people are actually responding properly. So whether or not either there is an improvement in phenotype or there is still the same control phenotype, but a phenotype that requires less medication to maintain. We can divide the subjects into either responders or non-responders to the fasting intervention, and we can see what characterizes these subjects already at baseline, trying to see, is there something that can let us personalize this? Can we measure stool and immune system from serum in a subject already at baseline and determine whether or not they're likely to gain a better cardiovascular phenotype by undergoing a fasting intervention? And it turns out that there are a number of the features that we saw changed by the fasting that actually seems to be perturbed at baseline. Specifically, most interestingly, I think, is the propionate production that I mentioned. So the subjects most likely to benefit by getting reduced hypertension, reduced need for hypertensive medication by a fasting regime like this are the ones that were depleted prior to the study for the production of propionate, again, an anti-inflammatory bacterial metabolite in the gut. And those subjects then see an improvement happening during the fasting phase, which is, while it reverts 3 months later, it's been elevated for a sufficient time for them to shift somehow into a better homeostatic state. And of course, we want to follow this up in other cohorts. We want to follow this up with other dietary interventions, and we want to run the same kind of analysis on other types of cohorts as well, including drug treatment, including progression of disease. But the overall approach would be the same, to see which factors can we control for, which factors can we observe, can we find new mechanisms that we could potentially target directly. In this case, for example, one can consider probiotics and prebiotics to achieve the same thing. And can we determine factors of individual variation that help us assess which treatment is most effective for an individual from studies like this, getting closer to personalized medicine. So those would be a few examples of what I wanted to highlight, which is essentially multi-omics analysis of medical data, taking into account technical confounders, taking into account treatment confounders, both in a cross-sectional and a longitudinal setting, to try to build computational models for how health and disease emerges and try to improve treatment standards. A lot of people have been involved in this work. This is just a few examples of labs and individuals that were involved in the studies I mentioned. But I think I'll stop here and mention again, we are in Berlin. If you want to do postdoc projects with us, please contact us. And I will take any questions. Thank you so much.
Thank you, Dr. Sofia, for this wonderful talk. I'll encourage all the attendees to go and ask the questions. I can still read them. You can maybe stop sharing your screen if you want, as you want, if you still need the slides.
Yeah, I can open it up if needed, but I can see the chat if I shut it off.
Better.
Questions?
For me, I'll have a question. So first, it's an amazing data collection and I guess data cleaning included in this work.
Yeah, that's always an issue.
Yeah, I guess, and especially because you're taking a lot of different sources. I was asking myself, when you perform your association studies then on all these data, you were talking about going for the in vitro on the mouse, but based on key findings. But how do you define those key findings?
Well, it would make sense, ideally, for there to be some plausible mechanism for them. So if there's support from any other kind of experimental data that one can see a way why this would happen, that's one factor in their favor. But mostly from a data-driven approach, features that are significantly associated with the outcome in question, where there's no better explanation and where the effect size is large enough. Because of course, with enough data, with enough sensitive statistical methods, you can detect a lot of associations that achieve nominal significance even after controlling for multiple testing, but where the effect size, as in many GWAS studies, is so small that in practice it's not relevant for treatment choices. So I think those would be the criteria that I would mainly use. And then with more complex methods, such as building multi-step networks, looking for as direct connections as possible, or connections where we can also measure the intermediate stage.
Okay.
Examples here being things like the production of short-chain fatty acids, where we would also want to do things like direct infusion of short-chain fatty acids, or other ways of causing the same thing to happen.
Okay, and for example, when you were talking about the huge cohort, I think it was coming from Denmark, about 2,000 individuals. Out of these association studies, what was the range of power you managed to obtain?
When you say power, you mean effect sizes or a number of features?
The effect size.
Yeah, so generally it's quite subtle in terms of microbiome effect sizes. So I like this Cliff's delta measurement as a non-parametric equivalent of the Cohen's d for microbiome features, because then I need to assume less of the distribution. Typically things like the metformin versus E. coli association is an 0.4 effect size, 0.4, 0.6. So it's rarely very strong. It can be a little bit stronger for metabolite signals. So I think we have associations of elevated pyruvate in some heart disease patients that I think is up on like 0.6, 0.7, but it's rarely very strong. So it's more guiding us towards something than smoking gun in and of itself.
Yeah, for sure. And I had the last also question. So I really like all the analysis on the different collecting and the different how to say, the procedure to obtain data and to create them, basically. Are you already able to use these different comparison in order to guide people what to do and which extraction or which method to use in order to assess to go towards some kind of results?
Yes, we are doing this in Berlin now. For example, we're doing COVID microbiome studies where we have broadly recommended these room temperature stabilizer kits for DNA sampling, microbial DNA sampling, simply because it's so much more realistic to use them, because you don't need to drive out with dry ice and pick up the samples. But that was very much why we wanted to do that study, to make sure that it's justifiable to use these methods, and it seems that it loses relatively little signal. So currently we prefer the use of the SIMO kit, but I think there are competitors that use a roughly similar approach as well.
Okay, thanks.
I'd be happy to advise, by the way, if anyone wants to do a microbiome study, email me and we can send you our protocols. And this study is also out if anyone wants to read this in cardiovascular research.